Hi everyone, this is your instructor Kwame Shen, and this lecture is the final part of our lecture on prokaryotic classification. And in this lecture, we'll look at the class Gamma Proteobacteria and Delta Proteobacteria. And then we'll also look at other gram-negative bacteria that are of medical importance, just 3 different groups. Now, the gamma proteobacteria are the largest groups of proteobacteria. So you have the purple sulfur bacteria that belongs to this group. You have intracellular pathogens that are also members of this group. Now, for the purple sulfur bacteria, they are obligate anaerobes and generally would use hydrogen sulfur as the source of their energy. And so they're able to reduce hydrogen sulfur to sulfur, and they are typically associated with granules. And these granules may be deposited either internally or externally. Then you have intracellular pathogens with the genus Legionella and the genus Coxiella. The genus Legionella has members that cause Legionnaire's disease. Legionnaire's disease is mainly spread through mist, such as air conditioning. The bacteria is also known to live in water systems as well. And for a long time, it was very difficult to culture this type of bacteria. Then you have the Coxiella. The genus Coxiella has members such as Coxiella benneti. So the species name is benneti. So that's the species name, Burnettii, and which causes quarry fever or Q fever. It's called Q fever because for a long time we couldn't figure out what was actually the cause of it, just like saying X fever or Y fever. We didn't know what it was, but unlike other bacteria that looks very much like it, they can survive airborne transmission, which makes them unique and more pathogenic as well. Because they are intracellular parasites, as you can see here, you can see the bacteria living within the cells of an organism, and that's how the Coxiella benneti presents. When it's isolated, this is how it also appears. Then you have the methane oxidizes. So the methane oxidizes would live just above the methanogens. Remember, we talked about methanogens when we're talking about archaea. Now, the methanogens will produce the methane, and then the methane oxidizes will then use the methane as a source of their carbon or energy. And the genus Methylococcus is the representative of the methane oxidizers. Then you have the largest group of organisms under the gamma proteobacteria. They are called the glycolytic facultative anaerobes. So these organisms will typically use glycolysis. They are facultative anaerobes. So they would prefer an environment that has oxygen in it, but if there is no oxygen, they can still survive. Although they will produce energy at a less efficient rate, they will still be able to produce the energy. They may also use the alternative pathways the alternate pathways, such as the Ebden-Meyerhof pathway or the pentose phosphate pathway. So they typically rods, they may be curved rods. They typically will have flagella, some of them. And then you also have some of them that are cocci in shape. So this table, table 11.4, gives a summary of very important members of this group. So when we look at the family Enterobacteriaceae, which are straight rods, oxidase negative, they have members that are non-motile, but they are members that are also very motile, like the Proteus genera. So in this group, you have Exiraceae with the E. coli, the Enterobacter, Enterobacter aerogenes, Serratia marcescens, Salmonella, Proteus, Shigella, Yersinia, Klebsiella as well. And they cause different diseases. And so you have gastroenteritis that are connected to E. coli. Enterobacter and Serratia will rarely cause diseases, but you have enteritis, you have UTIs, shigellosis, plagues caused by Yersinia pestis, and Klebsiella pneumonia causing pneumonia. And then you have those that are vibro, curved, and also will have flagella, and that is the vibro genera. An example will be the ones that will cause cholera, vibro cholerae, which causes cholera. And then you also have the cocci or the straight rods that are non-motile. Example being the genera Haemophilus, which may cause meningitis in children. So you have very important pathogenic groups in this particular class, the gamma proteobacteria that are described as glycolytic facultative anaerobes. Within this group of gamma proteobacteria, you also have the Pseudomonas. The Pseudomonas, that's the group name. The other name is Pseudomonadales. So at this end, you have minus the S, you have ALES. But under this, you have members of the genus Pseudomonas, which is an important microorganism in the food industry, and also it causes UTIs. It is implicated in hospital-acquired or healthcare-associated infections. Example being Pseudomonas aeruginosa, which is very difficult to treat in hospital settings and easily transmitted to people who are immunocompromised. Then you have, among this group, you have the Azobacter and the Pseudomonas, which are also important members of this group. So these are free living in the soil. So they live freely in the soil, free living. And it's in this state that they are able to fix nitrogen. They're typically not associated with the roots of plants, unlike the Rhizobium and Nitrobacter species that we looked at earlier. So Pseudomonas would typically appear in pairs, as you see in this picture. So they will lie side by side in pairs, and you'll see that they have their flagella, their polar flagella, unipolar. The smallest group among the proteobacteria is the delta proteobacteria. So among this group, you have representatives such as the salvo-vibrio, which will convert sulfur to hydrogen sulfide. Hydrogen sulfide will then react with iron to create that black precipitate that we see. And then you have Delovibrio, and Delovibrio will prey on other bacteria, other gram-negative bacteria. Very, very interesting. And then you have myxobacteria, which typically dwells in the soil, but could also prey on other bacteria as well. So we'll look at their life cycles in brief. For Dilovibrio, so you have a Dilovibrio bacteria that is attacking the bacteria here. So this is the prey, and that's the predator. Once it enters the cell by digesting away portions of the cell wall and the periplasmic space, it gets in there and it starts multiplying, and then eventually it's able to consume the entire organism and then produce more offspring, lyse the cell, and then continue with the whole cycle. And so in this picture as well, you see the Dilovibrio attacking a bacteria that is going to be consumed eventually by it and use it to propagate itself. And then you have myxobacteria. Myxobacteria in its vegetative states will be able to multiply by binary fission when there's a lot of nutrients available. When nutrients is depleted, it glides and comes together to form a mass. And as it's gliding, it's producing enzymes. And any microorganism that it encounters, any bacteria that it encounters, it is able to digest it and becomes a source of food. Eventually, it develops fruiting bodies, as you can see here. So you have a fruiting body that's developed, and then it is going to produce spores called myxospores. The myxospores would then germinate once conditions are ripe, and then the cycle will continue. Then the last group among the proteobacteria in terms of the classes would be the epsilon proteobacteria. So these are rods, they are vibrios, and they are spirals. Important representatives would be the Campylobacter and Helicobacter bacteria. Campylobacter bacteria is associated with blood poisoning. Example would be the Campylobacter fetus. So C fetus. And C fetus will cause spontaneous abortion in domestic animals. And then you also have Campylobacter jejuni, which is an important foodborne disease agent. Then you have the Helicobacter, which are associated with cancer. So, for instance, Helicobacter pylori is a typical culprit in stomach ulcers. Pylori. So H. pylori. And you see pictures of these two groups of bacteria in here. You can see the curved nature, and you could also see the flagellum that are attached. Then there are other groups of bacteria that are of ecological or medical importance. So there are other different phyla, but we'll just look at the three groups that are of medical or economic ecological importance. So chlamydias is one group. They are cocci. They are very small in size, 0.2 micrometer, smaller than some viruses. They do not have peptidoglycan. They are intracellular, so they have to, just like rickettsia, they have to live, obligate parasites that will live within cells. But they are very important medical bacteria in terms of the impacts they have on us. So, for instance, if you take Chlamydia trachomatis, Chlamydia trachomatis is an organism that causes trachoma, which is one of the leading causes of blindness, especially in the developing world. And then they have species that are implicated in pneumonia. There are others that are implicated in sexually transmitted diseases. Example would be the lymphogranuloma venereum, which is a sexually transmitted disease. And that is how it appears as a granule in the pubic area, the pelvic area, and it's transmitted through sexual contacts as well. Then you have the spirochetes. The spirochetes are helical, they are corkscrew shaped. They live in diverse habitats, and their metabolism is also diverse. Important representatives will be the genus Treponema, where you would have Treponema pallidum, which causes syphilis. And then you also have another important genus, Borrelia, where you have species that causes Lyme disease. And so these are transmitted by ticks. So in the western United States, especially in Southern California, the western black-legged tick, also known as Isodis pacificus, is the vector of this disease. Then you have the bacteroids. The bacteroids are obligate anaerobic, rod-shaped, and they're able to catalyze complex carbohydrates. So you see them commonly in the digestive tract of mammals. Up to about 1 billion per gram of feces of humans are filled with these particular bacteria, and they are typically recovered from deep tissue infections or surgical punctures, and so they can also cause such infections as well. So you see the spirochetes in this picture here, helical, corkscrew shape, and then the bacteroids as well. So this is the last group of the bacteria that we're looking at under classification. And so that brings us to the end of our classification of prokaryotes, which covered archaea and bacteria.